Welcome to the Crazy Alchemist podcast, where alchemy, occultism, and strange history come alive.
What do you think the lab would find if Jesus' DNA could be tested today? Sit with that question for a moment. Pick an answer in your head. Then I'll walk you through what each possible answer would actually mean for an atheist, for a Christian, for a Muslim, for a Jew. The piece commits to no answer of its own. Material from Aquinas in 1272, the Quran in the 7th century, the Talmud in the 3rd to 6th centuries, in the lab papers from the last 40 years, all sits inside each case where it becomes relevant. Before we get to the cases, I want to set up the basics. What does a modern ancestry testing lab actually measure? A modern ancestry testing lab reads three things from a sample: mitochondrial DNA, inherited only from the mother in an unbroken matrilineal chain back into deep prehistory, the Y chromosome, the smaller of the two sex chromosomes, present only in males, inherited only from the father, and the autosomes, the other 22 pairs of chromosomes, recombined in every generation from contributions by both parents, mother's line, father's line, and the recombined mix. For any biologically conceived human male, the three slots fill in cleanly and the lab assembles a report. For Jesus, that is where the question begins. Mary's half is the easy side. The mother's contribution is the part nobody disagrees about. A first-century Galilean Jewish woman would carry mitochondrial DNA from a regional maternal lineage, almost certainly some subclade of haplogroups H, J, K, T, or U, the dominant maternal lineages of the Iron Age and Roman period Levant. Her autosomal contribution would cluster with what modern population genetics calls the Levantine reference group, closest living relatives in modern Lebanese Christians, Druze, Samaritans, and Mizrahi Jews. The continuity from Bronze Age Levant to today is well documented in modern genetics, so Mary's half of Jesus' DNA is, on every theological reading, ordinary first-century Galilean. The atheist, the Catholic, the Eastern Orthodox, the Muslim, and the Jew all expect the same result on the maternal side. The question was never about mum. Now I want to introduce one idea before we get to the cases, because it's the central idea that holds everything together. DNA, in a literal sense, is a document. There is no such thing as generic local DNA. A lab result like Levantine male JP58 describes a document. Think of it like a passport full of stamps from countries you've never visited. Each point mutation along the Y chromosome's history corresponds to a real man who lived at a specific time, carried that specific mutation, and had a son. A JP58 chromosome encodes the patrilineal record of a chain of ancestors stretching back roughly 3,000 years. If God produced a Y chromosome for Jesus that reads as an ordinary Levantine J1 or J2 chromosome, the chromosome would be encoding a paternal lineage that, on the Christian and Islamic theological accounts, did not exist. Each mutation would be a fictitious ancestor's claim to existence. The cellular machinery would read a document about people who never lived. A Christian apologist could push back here. Perhaps God copied the exact Y chromosome of King David and gave it to Jesus, in fulfillment of the prophecy that the Messiah be of David's seed. The result would still record ancestors who lived 1,000 years before the conception, and through whose bodies Jesus did not biologically descend, and the divine actor would still be the source of the chromosome instead of a biological father. The objection sharpens. Forgery or duplication, the document records a lineage the body did not run through. A second apologist move appeals to divine accommodation. God works within human frameworks of understanding, the argument goes, and producing a Y chromosome that reads as an ordinary human lineage is part of meeting humans where they are. The question raised here is whether creating a false historical record, even for a purpose, differs meaningfully from deception. One philosophical assumption is doing all the work in this thought experiment. It binds theology to biology and treats the lab's answer as evidence about the doctrine. A believer can always step outside that frame, since a miracle, by definition, operates outside ordinary biological mechanism, and a result that contradicts the doctrine biologically does not have to contradict the doctrine theologically. The test only has bite if you assume that reality is biologically consistent, even where the divine acts. So with that setup in mind, let me walk you through the 5 possible reports the lab could return. I'll take them one at a time. Case 1 is the simplest result. The lab returns DNA from a first-century population in plausible contact with Galilee. Y haplogroup J1 or J2, autosomal matches closest to modern Lebanese Christians, Druze, Samaritans, and Mizrahi Jews. A perfectly ordinary first-century Levantine Jewish male profile, or perhaps a Roman soldier's profile, or a Greek trader, or an Anatolian, an Egyptian, a Nabatean. The first-century Levant was a meeting point for a dozen population streams.
What does Case 1 mean for the historical critical reader, the reader who treats the Gospels as historical documents to be analyzed? This is the expected result. Joseph, or some other first-century man, was the biological father. The virgin birth was a theological claim made about an ordinary biological birth. There's a sharper version of this position worth naming. Paul's letters, written in the 50s of the first century, do not mention a virgin birth. Mark, the earliest gospel, written around the year 70, has no nativity story at all. Only Matthew and Luke, written around the years 80 to 90, include the virgin birth, and the 2 genealogies they offer for Joseph contradict each other on most names between David and Joseph. Matthew traces through Solomon, Luke through Nathan, both supposedly along Joseph's line. On the standard historical critical reading, the virgin birth is a late accretion that the earliest Christian sources do not know. What does Case 1 mean for the Christian? This is the result most Christians expect without thinking, and it forces the deepest reinterpretation of the doctrine. The virgin birth either has to be read as theological language that does not bind biology, a move some liberal Protestants have already made, or accepted alongside a forged biographical document encoded into nucleotides. The forgery reading sits uncomfortably with the classical monotheist commitment that God does not deceive. The Hebrew prophets required the Messiah to be of the seed of David. Both gospel genealogies trace through Joseph. If Joseph is not biological, the Davidic claim is legal only. There's a sharper version of the Davidic problem inside the Hebrew Bible itself, and this is where the conversation gets historically interesting. It's known as the Jeconiah curse. Jeremiah chapter 22 verse 30, addressing the Davidic king Jeconiah before the Babylonian exile, says, "Thus saith the Lord, write ye this man childless, a man that shall not prosper in his days, for no man of his seed shall prosper, sitting upon the throne of David and ruling any more in Judah." Now Matthew's genealogy in chapter 1 verses 11 and 12 drops Jeconiah straight into Joseph's family line. The early rabbis had a way around the curse. They read it as lifted when Jeconiah repented during the Babylonian exile. The Talmud, in the tractate Sanhedrin, builds the case through a small Hebrew wordplay on the name of Jeconiah's son, Shealtiel, reading the name as God consulted his court and freed him from the oath. For the rabbis, the curse was over.
Medieval Jewish writers then turned the same curse against the Christians. Either Joseph was the biological father, in which case Jesus inherits the curse and cannot sit on David's throne, or Joseph was not the father, in which case the whole Davidic claim through him collapses. The argument circulates in the medieval Jewish polemical literature of the Disputation period and is in the same key as the arguments the Rabbi Nachmanides put to a Christian opponent at the Barcelona Disputation of 1263. The shape of the objection is older than the Gospels. The genetic vocabulary is just the most recent layer of an old conversation. What does Case 1 mean for the Muslim reader? Quran chapter 3, verse 59 explicitly parallels Jesus' creation to Adam's. Indeed, the example of Jesus in the sight of Allah is like that of Adam. He created him from dust, then said to him, "Be," and he was. The classical commentary, including Tafsir ibn Kathir, reads this as parallel miraculous creation by direct divine command. Case 1 contradicts the verse on a strict reading, since a J1 or J2 Y chromosome implies a human father in the ordinary patrilineal sense. The verse can be read in three ways under Case 1. As metaphor that does not bind biology, a reading some modernist Muslim thinkers might allow; as a claim about outcome over mechanism, since the phrase "be" and "it is" gives Allah the power to create any Y chromosome by direct command, including one indistinguishable from Joseph's, or as literally incompatible with a J1 or J2 result. Mainstream Muslim scholarship has not, as far as the published literature shows, worked through which reading the verse requires. For the Jewish reader, Case 1 is the expected result, and it returns the messianic question to its older form. The Jeconiah curse problem is real if Joseph is biological. Other messianic criteria remain unmet anyway. He did not establish a political kingdom, did not gather the exiles, did not bring universal peace. The genetic test settles only one branch of a multi-branch question. That's Case 1. On to Case 2. Case 2 is what happens if the lab returns DNA from a population that could not have been in plausible first-century contact with Galilee, a pre-Columbian American lineage, an Australian Aboriginal one, a Polynesian people of the deep Pacific. Case 2 mostly sharpens the forgery problem from Case 1. The historical critical reader's confidence drops since the naturalistic explanations are thin. The Christian faces a forgery reading where the forged lineage points at a population with no narrative role in the Christian story. The Muslim reading of 359 still asks for a Y that looks like nothing on the human tree. A real population in an impossible location does not satisfy that prediction. The Jewish messianic frame has no category for descent from a population unknown to the prophets.
The one philosophical wrinkle in Case 2 is the migration possibility. Maybe a Phoenician trader reached the Americas, or a Polynesian voyager reached the Levant, and the archaeology has not turned it up yet. The result would then slide from definitively supernatural toward definitively rewriting human migration. The difference between those two outcomes might be smaller than it sounds. Now, Case 3. This is where things get strange. Case 3 is what happens if the lab returns DNA that matches no human population at all, a Y chromosome that does not sit on the human phylogenetic tree, with conserved regions intact so the cellular machinery can still use it, but variable regions matching nothing in any extant or extant human population. For the historical critical reader, this would be the most extraordinary single finding in the history of genetics. The naturalistic options all have their own problems. A Y chromosome with sequences matching no extant or extant human group, no archaic hominin like Neanderthal or Denisovan, and no close primate would be the most difficult result for biology to contain within current naturalistic frameworks. The honest historical critical position would settle on currently unexplained. Reaching for supernatural is a stronger claim than the result would license. For the Christian, Case 3 is a reading that bypasses the Davidic and forgery problems by removing the human paternal lineage altogether. There is no chain of fictitious ancestors to forge, and no Joseph through whom the Davidic genealogy has to run. Some early Christians actually held a related view. Arius of Alexandria, condemned at the First Council of Nicaea in 225, taught that the Son was created by the Father and had a beginning in time. An Arian framework would absorb Case 3 with no contortion, since Jesus would be a special creature brought into being by direct divine action. The post-Chalcedonian framework that became orthodox has more work to do. The Council of Chalcedon in 451 declared Jesus homoousios, of the same substance, with us according to his humanity. A Y chromosome created from nothing for the incarnation complicates that claim, since the genome would not biologically descend from any human lineage. For the Muslim, case 3 is what Quran 3:59 predicts taken seriously as a biology claim. The verse parallels Jesus' creation to Adam's, and a Y chromosome with conserved deep mammalian biology in the variable regions outside the human tree is what the Adam parallel implies, if the verse is read as a biology claim. Whether 359 is meant as biology or as metaphor is itself contested in classical and modern Muslim commentary. No major Muslim scientist has worked the prediction out in modern genetic terms.
For the Jewish reader, case 3 is a problem of a different kind. Jewish messianic expectation has always been more this-worldly than the Christian. A divine Y chromosome doesn't fit the Jewish messianic frame, which expects a political king of David's line, biologically Davidic, who establishes peace and ingathers the exiles. Case 3 satisfies neither the genealogical requirement nor the political one. One quick caveat on case 3. It is among the most improbable of the 5 in the strict statistical sense. A Y chromosome that doesn't match anything is one specific result among billions. Any reader of any persuasion can ask, is this the expected result because it is wanted, or because the evidence points there? Case 4. This is, in my view, the most fascinating one, and it's the one believing biologists have written about most. Case 4 is what happens if there is no Y chromosome at all, an XX karyotype with miraculously activated SRY, a chromosomally female karyotype with the SRY gene translocated onto an X chromosome, producing a male phenotype despite an XX karyotype. Mary is the sole genetic source, the Holy Spirit as the activator that flips on male development. I want to start with the plain version of what this would actually mean for Jesus' body before any tradition's reading. People born with XX male syndrome develop male anatomy, grow up identifying and living as men, and are typically diagnosed only when they discover they are infertile or get a karyotype done for an unrelated reason. From the outside, they look like any other man. The lab report is the only place the condition shows up. So case 4 means a Jesus who looked male, walked through Galilee as a man, was crucified as a man. The lab report would have shown 46XX, where a doctor would normally expect 46XY. The depictions in art, the gospel descriptions, the iconographic tradition all stand. They track phenotype, and the phenotype is male. For the historical critical reader, case 4 is implausible but not theologically loaded. XX male syndrome is a known rare condition, occurring in roughly 1 in 20,000 male births by ordinary natural mechanisms. For Jesus, with no biological father, the SRY gene would have had to arrive by some other route. The historical critical reading would treat the result as a fluke, or as evidence that Jesus had an unusual biology, not as evidence for the supernatural per se.
For the Christian, especially the Catholic, case 4 is what Aquinas' Summa Theologica predicts when you translate his theology into modern biology. Back in the 1270s, Aquinas asked a simple question, whether the physical matter of Christ's body should have been taken from a woman. His answer? Although the Son of God could have taken flesh from whatever matter he willed, it was nevertheless most becoming that he should take flesh from a woman. Then, a few articles later, Aquinas makes a careful distinction between the source of the bodily material and the cause of the conception. The matter of Christ's body is taken entirely from Mary. The Holy Spirit is the active or efficient cause of the conception, but does not supply biological material. In modern biology, that translates almost exactly to parthenogenesis from Mary's egg with a miraculous activation step. The result in modern biology is 2 copies of an X chromosome and no Y. Jesus would be an XX male. There is one biological caveat to add here. The SRY gene, the gene that triggers male development, lives on the Y chromosome. If Mary contributes only X chromosomes, there is no SRY in her genome to activate.
so the divine cause has to synthesize the SRY sequence, about 828 base pairs of DNA encoding a 204 amino acid protein, and place it on one of the X chromosomes. The document problem from earlier doesn't quite return for SRY. SRY is a functional gene. It does not encode the historical patrilineal record that a full Y chromosome encodes. A wild-type SRY synthesized by the divine cause would code for the male development switch without fabricating any ancestors. Of the 5 cases, case 4 requires the smallest divine genetic intervention. The Catholic doctrinal commitment is more comfortable with this case 4 reading than most Catholics have been told. The art and the iconography are unaffected. Jesus would have looked male, and the depictions would have been accurate as records of his appearance. The shift is at the chromosomal level and in the doctrinal claims that rest on it. The Catholic argument for an all-male priesthood in particular has often appealed to Jesus' maleness and his choice of male apostles. A case 4 result would force a more careful definition of what male is doing in that argument. Phenotype and identity, which Jesus would still have had, or chromosomes, which he would not.
A quick note on the science behind this. An American Christian biologist named Kessel first proposed a related mechanism back in the 1980s, suggesting parthenogenesis from Mary's egg followed by natural sex reversal, producing a chromosomally female Jesus with a male phenotype. A British geneticist named Berry sharpened the idea in the 1990s, bringing modern genetics to bear on the XX male and SRY translocation mechanism. And just last year, in 2024, a team published a book with Cambridge University Press called Jesus and the Genome, the most established academic engagement with the question to date. We'll come back to this small body of work in a minute. For the Muslim reader, case 4 is incompatible with the Adam parallel. Adam, in Islamic tradition, was fully male, created from dust, not from the doubled material of any other human. A doubled Mary genome with translocated SRY does not fit the Adamic creation pattern. For a modern reader thinking about gender, case 4 is worth sitting with. The most worshipped figure in Western history would have a chromosomal karyotype most people would call female in a body that looked, lived, and was treated as male. Modern medicine groups XX male syndrome under the category difference of sex development, a category of natural variations documented in human biology. The intersex reading sits inside the doctrine. Take Aquinas' account of materia and the spirit's active cause seriously as biology, and an intersex outcome is what the doctrine predicts on its own terms. None of the Church Fathers anticipated this reading because the chromosomal vocabulary did not exist. Aquinas anticipated the structure of it without knowing what the structure implied. One last thought on case 4 before moving on. Case 4 is the case the believing biologists have written about most directly, and it is the one that quietly translates Catholic doctrine into modern biology more closely than the doctrine's own institutional formulations. The repeated independent landings on the XX male mechanism might mean it is the true reading. They might also mean that several biologists worked through the same logic and reached the same answer because there are only so many biologically possible answers under the doctrine. There's a third possibility worth flagging too. Case 4 feels more complete than the others because biology and theology happen to align cleanly here, and the cleanest alignment is also the most tempting reading. Elegance is sometimes truth and sometimes preference. And finally, case 5, the most extreme of the 5. Case 5 is what happens if the lab returns DNA that matches no human population on either side, maternal or paternal. The Y chromosome, the mitochondrial DNA, and the full autosomal pattern all return as sequences that do not match any extant or extinct human population. Mary did not contribute biological material in the ordinary sense.
For the historical critical reader,
Case 5 is biologically impossible by any naturalistic mechanism. An egg has to come from somewhere, and mitochondrial DNA is inherited from the cell that contains it. If the mitochondrial DNA matches no human, either the egg was not Mary's, which contradicts the gospel narrative, or the egg was created from scratch. For the Christian, Case 5 vindicates a very old and mostly suppressed reading. The early Gnostic Christians, particularly the Valentinians, taught that Jesus passed through Mary as water through a pipe and was not made of her flesh. Tertullian condemned this view in a work called On the Flesh of Christ, written around the year 206, precisely because it threatened the doctrine of the Incarnation. A Case 5 result would mean the Gnostics were right and Tertullian was wrong. Mary was a vessel, not a source. The Catholic title Theotokos, meaning God-bearer, defended at the Council of Ephesus in 431, would have to be rethought. Aquinas' Question 31, Article 4, which said the matter of Christ's body came entirely from Mary, would be falsified. For the Muslim, Case 5 fully satisfies the Adam parallel of Quran 3:59. Adam had no mother and no father, and Case 5 makes Jesus parallel in both directions. Mary's role in Surah Maryam, chapter 19 of the Quran, would have to be read as a sign attached to the moment of birth, with no claim of biological maternity attached. For the Jewish reader, both biological lines are gone. There is no genetic descent from David through either parent, and whatever Jesus was on a Case 5 result, he was not what Jewish messianic expectation was looking for. One practical note on Case 5. 2 parents' worth of DNA both matching nothing is a higher hurdle than 1. The first thing every biologist would do with this kind of result is check for contamination or methodological artifact. Even after that, real would mean only that no one can yet explain it. So those are the 5 cases. Now I want to step back and look at the surrounding territory.
First question, who has actually tried to answer this? Almost no one in mainstream science. Over the last 40 years, I could only find 5 serious attempts. You have an American biologist in the 80s who first proposed the sex reversal idea, a British geneticist in the 90s who sharpened it. There's a fascinating, if fringe, paper by a Turkish Muslim embryologist proposing that Mary herself was a chromosomal chimera with both ovary and testis. A Latter-day Saint biologist wrote a book on the chromosome question in 2022, and finally, just in 2024, a team published the Cambridge University Press book Jesus and the Genome, the most established academic engagement on this to date. The volume of work is small, and the distribution of who has written it is unusual. None of the central cohort of believing biologists most readers might name has published a sustained biological analysis of what Jesus' hypothetical genome would contain. That 2024 Cambridge book is the first signal that this might be changing. Now, before we wrap up, I know what some of you are screaming at your steering wheels right now. What about the Shroud of Turin? Don't we have actual physical relics we can just test? The honest answer is that the relic record is thin. 3 independent labs carbon-dated the Shroud of Turin to between the years 1260 and 1390. Casabianca and colleagues challenged that result in 2019 on statistical grounds when the original raw data finally became public, and De Caro and colleagues challenged it in 2022 on a novel X-ray dating method. Neither paper has overturned the medieval reading. Barcacia and colleagues recovered DNA from the shroud in 2015 and found a multi-century mixture of 14 mitochondrial haplogroups belonging to people of many origins who handled the cloth, not Jesus. The Sudarium of Oviedo's AB blood claim sits in non-peer-reviewed conference proceedings. The 1971 Lanciano myocardium study was single investigator with old blood typing methodology and no DNA work and no independent reanalysis in the half-century since. The Sokolka and Buenos Aires Eucharistic samples never reached peer review. So here's where we land. The body will not be found. The test will not be run. The lab report exists only as a thought experiment, and the thought experiment has already done its work.
Each reader holding the question has to admit something about the position they walked in with. The materialist gets the expected result in case 1 and has to confront something biologically unexplainable in any of the other 4. The Christian who accepts case 1 has to face the Jeconiah objection that the rabbis raised at Barcelona in 1263, an objection that sequencing only sharpens. The Christian who hopes for one of the other 4 cases has to choose between a forged biographical record and a Gnostic reading the Church spent centuries trying to bury. The Muslim has to decide whether Quran 359 was always a biological claim or always a metaphorical one. The Jewish reader can look at all 5 reports and find that only case 1 preserves the genealogical possibility, and even there, the political messianic criteria the prophets wrote down remain unmet. And here's the closing thought. The test would reveal, on any of the 5 outcomes, the framework each listener brought to it. The DNA does not arbitrate between frameworks, it exposes them. The question of what counts as proof of God is older than DNA and would not be settled by it. So which of the 5 cases did you pick in your head at the beginning of the episode, and did this change your mind? Let me know in the comments. If you enjoyed this, look for The Crazy Alchemist on YouTube and crazyalchemist.com for more. Thanks for listening.
If you enjoyed today's episode, make sure to visit crazyalchemist.com for more intriguing content and follow Crazy Alchemist on Instagram and Facebook to stay connected.